Hi everyone, welcome to the beginning of Chapter 5, where we talk about biomolecules, which are among the largest structures found in a cell, outside of organelles, if you will. But organelles are combinations of biomolecules. So these are the real structural and functional components of a cell. We've gone through the chemistry and some of the aspects of subcellular function. Moving into now, we're going to build biomolecules, and then in Chapter 6, we're going to get into organelles. And so these are the molecules of life, the biomolecules. Many of them can assemble into polymers. And so we have proteins, lipids, carbohydrates, and what we call nucleic acids, which is primarily DNA and RNA. They are large and complex much of the time. Some are relatively simple. Like for instance, a simple sugar is relatively simple and it's still used as a biomolecule, but we can form polysaccharides like glycogen and so forth that are very, very complex. And they do have unique properties. Again, that carbon backbone with functional groups and maybe the arrangement of the carbon atoms and so forth give these structures their unique chemical and functional properties. In many cases, understanding the structure gives us insight into how to handle diseases, how to handle issues when the structures aren't correct. So understanding these structures has been a major push over the last decade or so, and more sensitive techniques have come along. Just to illustrate, this is a structure of what's called the spike protein. The membrane would be up here, and I'll show you another picture here in a second. But with predictive analysis, they can see how the spike protein can open up and interact with an enzyme inside our lungs called ACE2. So we can actually begin not only to see the shape, but how this molecule can behave in the situations when we're talking about an active protein situation. Another image of this is that same spike protein, only it's showing with the membrane here. So you can actually see some phospholipids. That's really cool. I haven't seen much of that recently, so they must be getting better with their predictive analytics and so forth. But this is the same protein. If we looked at the other one, you'd see that it would be opening up here in order to interact theoretically with the ACE protein that we find in our lungs and airways. And as I mentioned in the previous slide, there are a ton of career opportunities out here that are very, very lucrative and quite interesting from a technique standpoint, because you don't just sit in front of a computer. You have to crystallize these proteins, and that takes a very interesting set of skills. It's very much an art. So an artist may actually be successful in a career like this. But you don't have to be in a career in order to contribute. Your computer, while you're not using it, like my computer right here, is actually helping with the COVID situation. SARS-CoV-2 is another name for the COVID-19 virus. So it's looking at right now, just literally while I'm not working, it's looking at this polymerase, which is important for the viral genome to be replicated. It might be a really good druggable target to help block the ability of COVID to spread. So you don't have to be in a career. You can just have a computer and help out. Really neat stuff. The story with at least 3 of the 4 biomolecules, we also can call them macromolecules, is that they're polymers. They're repetitive chains of smaller individual building block monomers. So a monomer is the building block, and we link the monomers together to form these complex structures. As I mentioned, 3 of the 4 classes are specifically this way. Lipids, it's a little bit of a more complicated story, but certainly for carbohydrates, for proteins, and for nucleic acids like DNA and RNA, this is the story of monomers being assembled into polymers. The synthesis and breakdown of these polymers, specifically proteins, carbohydrates, and nucleic acids, use a common chemical mechanism. They use different enzymes because the enzymes are specialized for a given type of polymer, and monomer reaction. But these enzymes all use an identical chemical reaction to do this. When we're assembling monomers together and we're adding monomers on to lengthen a polymer, that reaction is called a dehydration reaction, sometimes called dehydration synthesis, where we are putting two monomers together, or we're tacking a monomer onto the end of a polymer. And when we do that, we create a water molecule that is a product of that reaction. On the other hand, if we are breaking polymers down into monomers, that reaction is known as hydrolysis, or you could say hydrolysis. Lysis means destruction. Hydro means that we are using water, and then water is incorporated into the monomers, so that the water is itself a reactant in this and is destroyed in the reaction because an OH goes on one molecule and an H goes on the other. So we have hydrolysis and dehydration synthesis. But enzymes for different polymers and different molecules all use that similar process. Here at the top we have dehydration synthesis, where you see in one monomer there's an OH and in the polymer there's an H. When this bond is formed between 3 and the new monomer to become 4 here, we have that water produced. So water is a product of dehydration synthesis. When we're doing hydrolysis, when we're breaking down a polymer and reversing this reaction, we're going to use a different enzyme. We're going to use water, and water is destroyed in the process because we put the H goes on one molecule and the OH goes on the other. And with polymers, you have the possibility for making many, many different structures with many, many different functions. Because if you have different monomers and the order and the spatial arrangement with which you assemble them, they can mean that you have lots of different macromolecules. And that's part of what we see in our variances across a population within a species or across species. These polymers can be made in virtually endless ways and create different types of functions in that diversity. So some of the natural polymers we see out there, for instance, silk and keratin, which is in our hair, very closely related, have similar properties. Collagen and gelatin, those are both a form of polymer, or elastin, actin, and myosin, all have polymeric structures. And their functions, they may be strong in a tension way, like for instance, you can pull on them and they will resist that tension. Other things like the gelatin other types of molecules resist compression in some cases. So your cartilage often has a lot of collagen and gelatin in it, and it's very good at resisting pressure. Instead of tension, it's resisting compression. Things like cellulose, your paper, is a polymer of sugar. Amylose, dextran, and chitin, the shells that we find in things like shrimp or crayfish or lobsters or crabs, that's made of a polymer of sugar. It's all sugar. And again, lots of different functions for different structures here. And these differences can make all the difference between a protein which behaves one way and a protein which behaves another, or whether a carbohydrate is digestible or not, whether a lipid is healthy for you or not healthy. And certainly the sequence of nucleic acids varies, and then that leads to different protein structures down the line. That's where we transmit our genetic information. So the sequence there is very, very important. So here's an example. Here's a starch polymer, and we have carbohydrate monomers, and there's a given bond between those monomers. Cellulose, which is very, very closely related to starch, look at though the arrangement of the monomers here. There's a slightly different bond here, which makes cellulose much, much harder to digest. In fact, most healthy people don't digest much or any cellulose at all. Doesn't mean that they can't. In fact, there's some recent evidence that people who are obese may have enzymes that allow for cellulose to be digested, which means more calories if you're eating a salad. If you can digest cellulose into sugar, then you can metabolize that sugar, and that's calories. So it's an interesting situation. Or for instance, glycogen, yet another bond structure here, and glycogen is a glucose storage molecule, usually found in skeletal muscle, but often found in liver as well. And here's polymer of DNA. We've already talked about that, so we can move on. So our first biomolecule group that we're going to drill down into is carbohydrates. Carbohydrates, we all know, and you've probably known since before grade school, that carbs serve as fuel. So everybody knows that. And trust me, if I ask you a question about carbohydrates, the last thing I want to know is that you know that it's fuel. So please don't go there if you have a question on carbohydrate function. It is much, much more importantly about the fact that it's a building material. It serves a very, very important cell recognition role as well. So they include sugars and polymers of sugars. The sugar that you're used to seeing on the table or in the kitchen is actually a disaccharide. It's made up of 2 monosaccharides. So monosaccharides are smaller. Doesn't mean the monosaccharides don't taste good. It just tastes a little different than the table sugar that you're used to having. That table sugar is called sucrose, and it's made up of a glucose and not a galactose, but it's another fructose, I think. Fructose, yeah. So they're macromolecules, they're polysaccharides, even our simple table sugar, and they can create incredibly complex structures. The bonds between the monosaccharides are known as glycosidic bonds. And we label the glycosidic bonds, and this is, again, these are the bonds made, these are covalent bonds made by dehydration synthesis. The numbers and the alphas and betas have to do with what part of the monosaccharide is being linked to another part. And that's where the digestibility comes in, and sometimes the strength, depending on the type of glycosidic bond. Again, a covalent bond made by dehydration synthesis between carbohydrate monomers. To show you how important carbohydrates can be as a structural protein and as a signaling protein, above and beyond that fuel thing. I know fuel, it's great to know that you know it's fuel, but that's just we're beyond that now. Carbohydrates are a major component in what is known as extracellular matrix. Extracellular, outside the cell, material found outside the cell. Like for instance, if you touch your skin right now, you're touching dead cells that have lots and lots of keratin in them. And the keratin has lots of carbohydrates associated with it. That's why it can be kind of waterproof and relatively crystalline in structure. So all you see, these little kind of bottle brush looking things, these are carbohydrates that are attached to proteins and so forth. In the case of a heart, the muscle is called myocardium. If you have a heart attack, a myocardial infarction, not only are the cells screwed up, but the extracellular matrix also gets screwed up as well. And so maybe some cells die, but maybe there are some cells left. Even the cells that are left behind sometimes have trouble because the extracellular matrix is no longer arranged the way it should be. And that's leading to responses in those cells that is not as functional as original healthy heart tissue. So carbohydrates are fundamentally made up of this ratio here, 1 carbon for 2 hydrogens and an oxygen. When we're talking about just straight up carbs. Monosaccharides, like for instance glucose, has a 6-fold version of that. So we have 6 carbons, 2 times 6 is 12, and then 1 times 6 is 6. So we have C6H12O6. That's the most common monosaccharide, but we have a whole host of others. But they all still have this same ratio of 1 to 2 to 1. They're classified by where certain functional groups are located and the number of carbons in the carbon skeleton. Like for instance, we have pentoses and hexoses and other types of carbohydrate monosaccharides that are variations. Like for instance, in DNA and RNA, you have a sugar called a ribose sugar, which is 5 carbons. So we see here some examples: 3 carbon sugars, doesn't even call it a sugar. They call it a glyceraldehyde or dihydroxyacetone. That's because of the functional groups in it. So these are 3 carbon sugars, and these are very common intermediates that we use once we've broken down glucose metabolically. Here's ribose. We talked about the ribose sugar, and here's another one, and here's fructose. Fructose and glucose make sucrose, so that's a double sugar. The thing I will also say, though, is that in biology, sometimes they show these structures like on standardized tests, but the truth is these things will actually spontaneously form rings. So this is one way of depicting them, but I prefer the ring structures when I discuss these molecules. So as I mentioned in the previous slide, I don't find these structures intuitively useful. And most of the time, if you're in biology anyway, you see them depicted in these ring-like structures, which are the functional structures by and large in biological systems, serving as the raw material for more complex carbohydrate structures. So here's the linear on the left and the ring form on the right. Most of the time I'm going to defer to this ring structure rather than the linear one. And sometimes we'll abbreviate, like for instance, we'll have a structure that looks like this. We don't need to depict all of the carbons and all of that. And so here, and the only way to make this a little bit better is to put an oxygen right at this point. So you hear there's an oxygen here. That's actually one of the tip-offs that this is a sugar, is that there's an oxygen right, in one of these parts of the rings. And in a ribose sugar or a deoxyribose sugar, that oxygen is right here, like where my pointer is right now. And so when we're linking carbohydrates together, whether it's 2 monosaccharides to form a disaccharide, or if we're adding monosaccharides onto a longer polymer carbohydrate, that's forming a covalent bond we call a glycosidic linkage or a glycosidic bond. For instance, if we're making a disaccharide maltose, we take 2 glucoses and we link them together, creating a water that's a product, and it's creating this glycosidic bond with this oxygen here. They call it a 1,4 glycosidic bond because in this ring, if the oxygen is the 0, if you will, then they have a carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, and then 6 carbon. So this one's at the 1, and this one over here, if we go to 0, 1, 2, 3, 4, that's why it's a 1,4 glycosidic linkage, which is a digestible linkage, thankfully. Fructose and glucose come together to form the table sugar that we're familiar with, sucrose. And again, we're going to do dehydration synthesis using different enzymes that are specific for these molecules. In any case, this one's called a 1,2 glycosidic linkage because of the carbon arrangement where the glycosidic bond is forming. Because we can harvest energy so well from monomer sugars like glucose, by linking them together, that means that we have a lot of energy available to us if needed. The structural roles is where I would rather us be. There are some very, very important structural roles that I want to talk about. But here's an example. So starch, a form of starch, is a polymer of glucose. So if we look at these 3 little rings right here, we're looking zoomed in here, and we have and what we can do is we have polymers of glucose, and what we can do is we can knock that, we can break that bond using a hydrolysis, and now we have something that we can use to build other things or break down to make ATP. Starch is a polysaccharide. We often see these in plants. Normally animals do not make starches. Animals make glycogen. So plants will store these starches and with these glycosidic bonds, and the simplest form is what we call amylose. It's a simple helical structure. But starch can get very, very complicated when we start changing the bonds and adding branches, and it can change the texture and even the taste in some cases. Glycogen, again, a different type of glycosidic bond, has a different behavior and serves as a storage molecule in muscle and certainly in liver. Here's cellulose. Cellulose is also a polymer of glucose, much less digestible with different kinds of bonds stabilizing it. Not only covalent bonds, but now we're bringing in some hydrogen bonds as well, which is part of why cellulose has more durability, can last longer in the environment than either glycogen or starch. And I'm sort of repeating myself here. This is just about glycogen zoomed in. It's mostly in liver and muscle. And when your blood sugar begins to get low, the muscle will begin breaking down the glycogen to sustain its muscular contraction. The liver will start breaking down glycogen to release glucose out into the bloodstream to sustain tissues that need sugar, like, for instance, your neural system. A little more about cellulose a little bit. It's usually tougher, it's more durable, and it has to do with not only the glycosidic bonds, but the hydrogen bonds. Now, how do we get to the hydrogen bonding ability? Why doesn't it happen in glycogen, but it does happen in cellulose, making it much more durable? It's because we're actually using two different forms that are closely related isomers of glucose. We have an alpha glucose and a beta glucose, and I'll show you that here in just a second. So here's an example of what I'm talking about, where isomers can make all the difference. So they have identical chemical formulas. This is alpha. What you'll notice is that we have the ring structure and that the oxygen, the OH here in blue, is on the downside of the plane. This thing is actually not very flat. It's actually sort of wrinkled like a potato chip or something. Nevertheless, this oxygen or this OH, this hydroxyl group on this carbon in this particular position will stick out a certain way. On the other hand, beta glucose over here, that OH, that hydroxyl group, is located in a different arrangement on that carbon, in a different position on that carbon, pointing in a different direction. And so with cellulose, what you have going on is that you're using a combination of alphas and betas. So here's our glucose polymer here. This is all alpha. That would be like glycogen. But with cellulose, what we have now is, look, instead of all of the the hydroxyl groups being on the bottom, now they're being flipped alternating, and that allows for hydrogen bonding. Just that one little shift in position is the difference between glycogen and cellulose. And we have enzymes that are very, very sensitive to this. So in animal systems, the enzymes will actually specifically make the alpha, but not much of the beta. But in plants, the plants have many more enzymes than humans do. And they have enzymes that'll make the beta glucose so that you can make cellulose. And I'm beating this to death a little bit, but because the reason we can't digest cellulose normally, now there are some people who are obese who seem to have that ability to digest more starch, which means more calories. Most people can't. And we call that fiber. So if you talk about dietary fiber, that implies cellulose in the food that we're eating. In the case of herbivores, what herbivores do is herbivores have a symbiotic relationship. They have a very, very large part of their digestive tract, which is analogous to our appendix. The appendix is actually is a vestigial organ in our digestive tract that was originally used to hold microbes that were able to digest cellulose. But herbivores like cows or termites and so forth, they often have high populations of these cellulose digesting microbes in their intestines, and that allows them to easily digest cellulose for energy. Another fun polymer of monosaccharides is known as chitin, and slightly different monosaccharide here. That chitin can form the shells, the exoskeleton of insects and things like lobsters and crabs, what we call arthropods. These things have a hard outer exoskeleton, if you will. And as they grow, they have to shed that old skeleton in order to grow a larger, newer one. Otherwise, they can't gain size. Very, very important structural for the endurable material, which makes it a wonderful material for surgery. So if somebody needs to have stitches way down inside them where it's very difficult to remove those stitches over time, a surgeon will use chitin uh, uh, thread. They'll use a chitin fiber in order to do those stitches, and over time that chitin will dissolve inside the tissues, hopefully after the tissue is healed. The tissues that the stitches that you have on the outside of your body are usually not chitin. They're usually some other type of polymer that's a little more durable. Chitin is less durable, but it works well because you don't have to go in and remove the stitches again. Okay, so moving on from carbohydrates, now we have lipids. Lipids are diverse in many ways, but the one thing that makes lipids a little different from the other three groups is that while they are made up of monomers, and you can actually polymerize fatty acids on these things, or you can have two here, or maybe you can change that head group and now you can have three. There are some polymer-like things, but it's less uniform, shall we say, than the other three groups. The main groups of lipids we have are triglycerides, phospholipids, and steroids, and maybe waxes are in there as well. But they're large. They can form large molecules that don't necessarily involve covalent bonding, so they're not true polymers. Yes, they have polymeric characteristics, but they're binding non-covalently in large molecules much of the time. So that's a little bit of a technical issue, but it's still accurate. They generally are hydrophobic, or if they have even a mild amount of they sometimes can be what we call amphipathic. That is to say, they can dissolve a little bit in oil and water. Like soaps are actually a form of lipid that allows for a little bit of mixing with water. But most lipids in our body are hydrophobic. They don't mix very well with water. Mostly hydrocarbons, a CH2 or ratio, shall I say. We'll talk about that more. So we have fats, phospholipids, and steroids. So my first picture is a little more involved, but these are kind of the big three. These are the ones that I will emphasize. Fats are one subset of lipids, sometimes referred to as triglycerides. That's because we have this backbone of glycerol, actually a form of alcohol, not the alcohol that you would drink. You'll find glycerol oftentimes in skin moisturizers and things. And then these are the more monomeric structures that can be added, subtracted, or they can be lengthened or shortened. We call these things fatty acids because at one end there's a carboxylic acid, but it's very repetitive and sort of angular. I'll show you some pictures of that. So we take the glycerol, add the fatty acids. So the glycerol has 3 carbons, which means we can add up to 3 fatty acids to it. If we add 3, that makes it a triglyceride. And there's that hydroxyl again. So we're using dehydration synthesis to create these fats. By linking the fatty acids and the glycerols, that's a dehydration synthesis. So we're adding it onto that carbon skeleton. So here's our fatty acid. This one happens to be saturated because every single bond in the fatty acid, again, here's the carboxylic acid right here. Every bond in that fatty acid, the carbon backbone, is a single bond. An unsaturated fatty acid, you'll have a double bond somewhere in there. We actually label these. Okay, if you've ever heard of omega fatty acids, omega 3s, that's because that's where the double bond is located. And if it's cis or trans, that can mean more or less of a kink. This one here is a cis unsaturated bond because this group and this group are on the same side of that double bond. If it was trans, we would flip that H in this remaining thing, and it would be on the same side. It would give it a different degree of kink. And so here we have glycerol, our 3-carbon alcohol. 3 OHs here, which means that we can link up to 3 fatty acids. This one is palmitic acid, just happens to be 18 carbons. Again, it's dehydration synthesis here. Not too worried about the ester linkage. That, enzymes can cleave those things selectively in some cases. But you can see the different length of fatty acids. Some are shorter, some are longer, and some may have kinks in them due to unsaturations. The more kinks you have, the more likely that fat's going to be liquid at room temperature. As I mentioned, fatty acids do vary in length, the number of carbons in them. and also in the number and locations or types of double bonds that they have. So here we have arachidonic and stearic and palmitic acids. Each of them are almost identical except for the length of the carbon chain in the molecules. If we have unsaturated fatty acids, they have at least one double bond, and the number of double bonds and whether it's a cis or a trans can affect the shape. Like these guys, these two here, oleic acid, which is often found in butter, has one double bond in it. Other ones may have more than one double bond, and which again gives it a distinct kinking, which can also affect its physical properties, its melting temperature, and how it can be metabolized in the body. And just another picture from the book for you. Then saturated fats can pack closely together using those hydrophobic interactions, which means that they're more likely to be solid at room temperature or slightly below room temperature. On the other hand, unsaturated fats will be liquid at room temperature, though if you have a really good bottle of extra virgin olive oil, you can often put that in the refrigerator and it'll solidify if you get it cold enough. But that kinking, in this case a cis double bond on that one fatty acid, is the major difference between this molecule and this one, and leads to major differences in their chemical properties. So we're hitting that 30-minute mark, but I'm just going to go, I've got a few more slides, and I think I can go through them relatively quickly. So hang with me here. I'm going to go just a little bit long. I already said this stuff, so I'm going to move on. As animals, we can synthesize our own fats. But most animal fats, with very, very rare exception, are saturated. But sometimes we need these unsaturated fatty acids for proper nutrition, like for instance in omega-3. Again, we call it omega-3 because it has an unsaturation. It has a double bond, 3 bonds from the very, very end, which we call the omega. Doesn't matter how long it is, as long as that bond is 3 from the end, it has very, very important chemical properties. And so we find different kinds of omega-3 fatty acids in different types of food, and they seem to help with protecting us against cardiovascular disease. So as you move into your adult lives, one thing you can do that's very, very easy and adds flavor and can often improve the quality of your food while still enjoying that goodness of fats, because fats taste good. Don't get me wrong, I like fats. In any case, you can use good fats to substitute bad ones, and that will help you have a healthier diet if you're going to have those calories, if you know what I mean. And so when we talk about triglyceride type fats, the fats that we're used to, yes, the major role is energy storage, but let's not diminish the life protecting functions that other things go on. So yeah, they will store them in fat cells, but it also cushions vital organs and serves a very, very important insulating role. So yes, while it is an energy storage, it will protect structures like brain tissue is made up with a lot of fat, eyes, arteries and so forth, from rubbing against other organs inside the body. So there's a very, very important protection role. Thermoregulation as well. There are certain types of what we call brown fats that are designed to generate heat when it's cold outside, and those vary if you live in a warmer or colder climate. Somebody I knew actually died because she had anorexia nervosa, and what happened was she lost so much weight that the cushioning fats between her aorta and her organs like her heart and her intestines went away, and it didn't come back. So even though she was able to overcome the anorexia and she gained a healthy weight back, she actually died of a syndrome because the cushioning didn't come back properly, and so she basically wore a hole in her aorta and died. Phospholipids are very, very closely related to fats, only this time they have a glycerol and only 2 fatty acid chains, sometimes that are saturated and sometimes unsaturated. In either case, though, that third spot there is for tacking on additional functional groups that make this head group hydrophilic. If these molecules are hydrophobic, and we use those in the lipid bilayer, they will tack on usually some other molecule onto that third position to make the head group hydrophilic. So the tails are hydrophobic, but the phosphate group and whatever is attached, functional groups attached to it, make it a hydrophilic head group. So here's a picture of one. This one's called phosphatidylcholine. It's a phospholipid, and the reason we call it phosphatidylcholine is here's one fatty acid, here's two. This is unsaturated. We put a choline molecule in that third position. So phosphatidylcholine is why it's called that. So the hydrophilic head group, hydrophobic tail group, and we can change that. It doesn't have to be choline here. It can be all sorts of things, 3, 4, 5 different types of head groups, and that can actually affect how the phospholipids behave. But again, the fundamental structure of phospholipids is that all those tails coagulate together to form a hydrophobic interior, and the head groups associate with water to form that hydrophilic shell, if you will, on the outside of those hydrophobic groups. The fun part about phospholipids is that if you add purified phospholipid to a watery solution, they will self-assemble. The hydrophobic interactions and the hydrophilic attraction will cause either small micelles to form. These are basically like a raspberry with a hydrophobic interior. Or it can form a liposome that has an aqueous interior, and this is a phospholipid bilayer type ball. Or it can form large sheets of phospholipid bilayer, depending again on the head groups and the fatty acid tails. So we can form this bilayer, and that's what gives the cells the plasma membrane with a hydrophobic interior and a hydrophilic surface on either side. That we call a leaflet, by the way. An inner leaflet that's hydrophobic and an outer leaflet that's hydrophilic. Depending on the phospholipid lengths and the head groups, you can get complicated interactions with phospholipid bilayers in the cells. So with lipids, we have the triglycerides, also known as fats. We have the phospholipids, and then we have what are called the steroids, which we've all heard that phrase from building up muscle. Well, steroids have one thing in common, and that is this 4-ring structure, A, B, C, and D. That is known as a steroid nucleus, and no matter what you tack on to the outside of it, if you have that 4-ring structure, very, very lipophilic, very, very hydrophobic, that's what we would call a steroid. So cholesterol, and that's what this molecule is right here, has a specific set of side chains that give it life-conferring properties. It's very, very important as a fluidity buffer in plasma membranes. It protects the membrane from freezing up if the temperature drops a little bit, and it prevents the membrane from dissolving if the temperature gets a little bit warm. It helps maintain the proper fluidity of the plasma membrane so that we can move things in and out of the cell properly. Cholesterol is also a precursor for other things like estrogen and testosterone and other types of steroids that we use in the body. Problem is, and we can make cholesterol on our own, we don't need outside help, but in our Western diet, because we eat a fair amount of meat and animal products, the cholesterol shows up, and if we have too much of it, your body ends up accidentally or otherwise shunting it to places where it coagulates and forms plaques inside the things like your blood vessels, leading to cardiovascular disease. There are different types of cholesterols. We're not going to go in there, but you should be able to recognize a fat, a phospholipid, and a steroid. So there are those carbon rings again. Again, this is the A and the B and the C and the D. Should be able to spot that from a mile away. I'm not going to ask you to identify what kind of steroid it is, but if you see that structure, you should know that it is in fact a steroid. And for our final slide today, I just have a couple of stick figures or space-filling models of cholesterol. Again, your goal with the lipids is to be able to identify the 3 categories: a triglyceride / fat, a phospholipid, which has 2 fatty acids and a head group, and then this steroid nucleus that we call a steroid. So here, as this thing spins around, here's A, And then next to A is B, and C and D. Promise me I won't give you a spinning one for your exam. A, B, C, and D through here. That red atom is the same in both of these. That's the OH of cholesterol. But you should be able to recognize cholesterol and know its two major roles: fluidity buffer in plasma membranes, and as a precursor for other steroid hormones like testosterone and estrogen and so forth. Oh, I'm going right at 40 minutes, so I'm going to end right there, and thank you for your time. We'll pick up part 2 of chapter 5 in the next video, where we deal with proteins and nucleic acids.